This video will discuss about the recombinant DNA technology. Our first topic is the basic technique for creating a genetically engineered bacterial cell. The gene is identified. Identifying a desired gene involves various techniques such as DNA sequencing, PCR, polymerase chain reaction, and gene expression analysis. Scientists often use specific probes or primers designed to target the gene of interest, allowing them to isolate, amplify, and analyze its sequence or expression levels. This process helps researchers understand the gene's function, structure, and potential application in fields like medicine or biotechnology. The gene is inserted into a plasmid. Inserting a gene into a plasmid involves using restriction enzymes to cut both the plasmid and the gene at the specific recognition sites. The cut gene is then legated into the plasmid, forming a recombinant DNA molecule. This process allows the gene to be incorporated into the plasmid circular DNA structure. The resulting recombinant plasmid can be introduced into hot cells, such as bacteria, for replication. This technique is widely used in genetic engineering to produce proteins, study gene function, or create genetically modified organisms. The recombinant plasmid is put into a bacterial cell. The recombinant plasmid is introduced into the bacterial cell through a process called transformation. Bacterial cells, usually Escherichia coli or E. coli, are made competent to take up foreign DNA. The competent bacterial cells are then mixed with the recombinant plasmid. This can be achieved through methods like heat shock or electroporation. The bacterial cell membrane becomes temporarily permeable, allowing the recombinant plasmid to enter the cell. Once inside, the plasmid can replicate along with the bacterial genome during cell division. As a result, the bacterial population would contain copies of the recombinant plasmid, and the gene of interest carried by the plasmid can be expressed or used for various purposes such as producing proteins or studying gene function. The second topic is placing together fragments of DNA from the different sources to produce a recombinant DNA molecule. Our restriction enzymes cut the DNA into fragments. Restriction enzymes, also known as restriction endonucleases, are proteins that recognize specific DNA sequences known as recognition sites or restriction sites. These enzymes act as molecular scissors, cutting the DNA at these specific sites. The recognition sites are typically palindromic, meaning they read the same backward and forward. When a restriction enzyme encounters its specific recognition site on a DNA molecule, it binds to that site and cleaves the phosphodiesterase bonds with the DNA backbone. This results in the DNA being cut into two fragments at or near the recognition site. The ends of these fragments can have specific overhangs known as sticky ends or blunt ends, depending on the type of restriction enzyme. This ability of restriction enzymes to cut DNA at precise location is crucial in various molecular biology techniques such as creating recombinant DNA molecules, gene cloning, and genetic engineering. A DNA fragment from another source is added. When DNA fragments from another source are added, it often involves using a technique called DNA ligation to join these fragments with a vector, such as plasmid. DNA ligation is a process where the DNA fragments are covalently linked together. Firstly, the DNA fragments, which may include a gene of interest or specific sequences, are typically generated through a process like PCR, polymerase chain reaction, or obtained from another source. These fragments are designed to have compatible ends with the vector or plasmid. The vector plasmid is also cut with the same or compatible restriction enzymes to generate ends that can be paired with the ends of the DNA fragments. The DNA fragments and the vector are then mixed together with an enzyme called DNA ligase. DNA ligases catalyze the information of phosphodiesterase bonds between the sugar phosphate backbones of the DNA fragments and the vector, creating a recombinant DNA molecule. This recombinant DNA molecule containing the gene or sequence of interest can be introduced into the host cells, often through a process like transformation in bacteria, where it can be replicated and expressed. The fragments stick together by base pairing. Base pairing refers to the specific hydrogen bonding interactions between complementary nitrogenous bases in DNA. In the context of DNA fragments sticking together, the pairing occurs between adenine and thymine, as well as between guanine and cytosine. When DNA fragments with compatible or complementary sequences are brought together, the nucleotide bases on one strand of DNA form hydrogen bonds. With the complementary bases on the other strand, adenine pairs with thymine through two hydrogen bonds and guanine pairs with the cytosine through three hydrogen bonds. These base pairing interactions are highly specific, ensuring that the correct sequence of nucleotides is joined together. In molecular biology techniques like DNA ligation, this base pairing mechanism is crucial for the successful joining of DNA fragments, allowing the formation of a continuous and functional DNA molecule such as recombinant DNA plasmid. DNA ligase pastes the fragments together. DNA ligase is an enzyme that plays a crucial role in pasting or joining DNA fragments together. It functions by catalyzing the formation of phosphodiesterase bonds between adjacent nucleotides in the sugar phosphate backbone of DNA. Here's how the process works. First, DNA ends with compatible overhangs. The DNA fragments to be joined usually have ends that are either blunt or have compatible overhang sticky ends. These ends are created by restriction enzymes during the DNA cleavage process. Second, binding of DNA ligase. DNA ligase recognizes and binds to the ends of the DNA fragments. It is essential that the ends are complementary to each other. Third, catalysis of phosphodiester bond formation. DNA ligase that catalyzes the formation of phosphodiester bonds between the adjacent nucleotides in the DNA backbone. It uses energy from ATP adenosine triphosphate to link the 3-hydroxyl OH group of one nucleotide with a 5-phosphate group of another, creating a continuous cell DNA strand. Fourth, resulting DNA molecule. The end result is a covalently closed continuous DNA molecule. In the context of molecular biology, techniques like gene cloning or creating recombinant DNA, DNA ligase is crucial for joining the gene of interest with a vector plasmid, forming a functional and replicable recombinant DNA molecule. This pasting process by DNA ligase is fundamental for creating intact and functional DNA molecules in various genetic engineering applications. Our last topic is the simplified process of recombinant DNA technology using the bacterial plasmid as cloning vector. A restriction enzyme cuts the plasmid is only one place, but cuts the human DNA molecule a thousand of sites. One of those fragments carries the protein V gene. The restriction enzymes cut the plasmid at a specific recognition site, creating a single cut site on the plasmid. However, when applied to human DNA, the restriction enzyme recognizes the uncuts at numerous sites throughout the genome, generating multiple fragments. If one of those fragments from human DNA happens to carry the gene coding for the protein V, it means that a specific fragment contains the genetic information required to produce protein V. In genetic engineering or cloning, scientists can isolate and extract the specific DNA fragment, incorporating it into a plasmid or vector. The resulting recombinant plasmid can be introduced into host cells, allowing them to produce protein V using the human DNA fragment as a template. Sticky ends of the plasmid match up with the sticky ends of the human DNA fragment according to the base pairing rules. The sticky ends of the plasmid and the human DNA fragment match up because of the base pairing rules. Adenine on one strand of DNA pairs with thymine on the other strand, and guanine pairs with the cytosine. This complementary base pairing ensures that the cohesive or sticky ends of the plasmid and human DNA fragment can hydrogen bond with each other, facilitating the successful ligation or joining of the two DNA molecules. DNA ligase joins the two DNA molecules, forming a recombinant DNA plasmid. DNA ligase joins the two DNA molecules by catalyzing the formation of phosphodiester bonds between the sugar phosphate backbones. In the context of genetic engineering, this process creates a recombinant DNA plasmid. The enzyme uses energy from ATP to seal the nick or gap between the cohesive ends of the plasmid and the human DNA fragment, resulting in continuous and functional recombinant DNA molecule. Under the right conditions, a bacterial cell takes up the recombinant plasmid. Under appropriate condition, a bacterial cell takes up the recombinant plasmid through a process known as transformation. This involves making the bacterial cells competent or capable of absorbing foreign DNA. The recombinant plasmid is then introduced to the competent bacterial cells, allowing it to enter the cells. Once inside, the bacterial cells machinery replicates the recombinant plasmid along with its own DNA. During cell division, this enables the reproduction of copies of recombinant plasmid within the bacterial population. Cell division results in many identical copies of the bacterial cell, all carrying the protein V gene. When these genes are expressed, multiple copies or protein V are made. During cell division, the bacterial cells containing the recombinant plasmid, which carries the protein V gene, replicate. This results in many identical copies of bacterial cells, all inheriting the recombinant plasmid, and consequently the protein V gene. When these bacterial cells express their genes, the cellular machinery reads the protein V gene extraction produce multiple copies of protein V, leading to the synthesis of the protein within the bacterial population.